Hi, I'm Joseph Duretti. I'm at the Department of Bioengineering at UCLA. I just want to thank the organizers for having me to be able to give a talk at this webinar conference. And I always like to start my talk with the fluorescence image that you see on the screen right here. So this is a fluorescence image of a sub-nanoliter droplets formed using only a pipette and microparticles. The objective of my talk is to convince you, the audience, that using this lab on a particle technology, you can enable really any researcher to perform a single cell secretion assays. So I originally came from a microfluidics background. And in this field, we've seen over the last really 2 decades or so that has been a very powerful tool, and that specifically it enables the creation of highly uniform compartments at the length scale of single biological entities. So this includes things such as proteins, DNA, as well as single cells, which I'll be talking about mostly in this talk. And the real strength of this technology is that it gives you the capability to take a heterogeneous, for example, cell population, be able to look at individual cells and study certain phenotypes, and then be able to identify, or in some cases even isolate, rare subtypes of these populations. And this has really been particularly powerful in areas such as the screening of CHO cells, B cells, and hybridomas for antibody production. This is of particular importance now, really, with some of the potential therapies for the COVID-19 being monoclonal antibody therapies. So being able to select out clones that are productive producers, for example, CHO cells, is a very useful technique. There's also other uses in areas such as doing functional T-cell analysis and screening, as well as CTC secretion profiling. And just to really kind of summarize some of the existing platforms out there, you have these kind of microstructured devices that contain the cells themselves. So for example, microchambers, or these kind of nanopens, some people call them, that use optofluidics to be able to manipulate single cells into these pens, microwell arrays, as well as microdroplets. I think it's important also to note that some of these platforms are more suited for the sorting application. So for example, the optofluidic platforms and the microdroplet platforms are a little more suited for doing actual sorting. And if you want to learn in a little bit more detail about some of these technologies, there's a great review from Rong Fan's group that came out last year discussing various microfluidic technologies for screening cells based off of functional secretions. Now, say you want to use some of these microfluidic platforms to do single cell secretion assays, whether it be in your lab or as a research project or as part of a company. So working in a microfluidics lab, you kind of see how a lot of these things actually work firsthand. So for example, you might see a paper where you have a nice clean microscope image of this microfluidic device that fabricates droplets. It looks really simple and straightforward and seems really just kind of easy to use. But realistically, we know that this is really a simple device embedded in a much larger chip. And if you wanted to fabricate one of these chips yourself with your own design, you need access to a high-tech cleanroom facility. Now, assuming that you have these things and you're able to manufacture a design that you want to use for your project, then to actually use these, in the most cases, you need access to specialized and often expensive equipment to actually run these devices. So this is several barriers of entry to actually using this technology. Now, say you don't want to go through all the troubles of making a custom chip, but you want to kind of explore the commercial options. And there are a number of commercial platforms that are available, and particularly for doing single cell antibody development. Microfluidics is one company. They have the Cytomine. This is a droplet-based platform for sorting and cells of interest based off their secretions. Berkeley Lights is another platform. This is utilizes optofluidics in order to look at cells and look at their production over time. Now, I have to say that these are very functional platforms that are really well at doing what they're developed to do. Now, I would like to kind of argue that there is a big kind of caveat for these systems. And just to kind of put that in perspective, I am currently at UCLA, which is one of the largest public institutes in the world. And we do not have access to either of these technologies, which to me means that a lot of people actually do not have access to these technologies, and that there is definitely a barrier to entry to for researchers to actually use technologies such as this. So although these are very capable platforms, I would like to argue that they are not yet ubiquitous, and not all researchers have access to these technologies. So the question I want to bring up then is really, so how do we create a technology that is accessible to more people to perform these assays? And to kind of rephrase that question a little bit, the question really is, can we perform these same single cell secretion assays, but using only common lab equipment? And what do I mean by common lab equipment? By that, I mean really common. So micropipetters, vortexer, centrifuge, well plates even. And this can also include more technically complex instruments. So things such as flow cytometers, for example. So flow cytometers have been around for multiple decades. They are well established in many research labs. Pretty much any research institute you go to should have some sort of flow core on campus with many of these flow cytometers available. Many companies have these, many clinical labs have these. So although it's more technically complex, it is a very accessible technology, and I would consider it common. So how could we perform this assay using these common lab equipment? Well, so let me kind of bring it down to what are the requirements that we would kind of necessitate to properly perform these assays. Well, the first thing we need to do is be able to create uniform compartments. So we need compartments so that we can isolate single cells, and also so that we can retain any secreted product with that specific cell. We need uniform compartments because if we're trying to differentiate, say, different cells that are producing different amounts of proteins, we need to make sure that the reaction volume is very similar in order to be able to properly differentiate their production rates. The second requirement that we need is ideally we would have some solid substrate to perform reactions. So this is sometimes can be something like a capture bead that has capture antibodies that specifically bind the proteins of interest. And then once you have that, the last thing you need to be able to do is to be able to read out and potentially sort in high throughput. So this could be through use of a secondary fluorescent tag specific to that protein. And as I was kind of alluding to before, if you want to do sorting, you'd want it to be compatible with a flow cytometer system. And so compatible, usually it dictates that it needs to be small enough to actually fit through the nozzle of the device, of the flow cytometer, so less than 100 microns. And it also needs to be in an aqueous phase. It can't be in oil, it can't be in any other system. So you can't put droplets through a standard flow cytometer. You need to have some other way of reading out and processing your sample. So our kind of solution that we came up with in our lab is this concept of lab on a particle. And the idea is quite simple and seems straightforward, but took quite a lot of time to develop. And the idea is that you could take if you take 3D structured particles that have unique shape and unique material properties, can you spontaneously create uniform compartments in order to isolate, say, in this case, for example, cells of interest. And any of these compartments or droplets formed by these microparticles, we refer to them as dropicles. And we've been developing this system over the last, I think, 4 years now. And here's just kind of a blanket view summary of these technologies. We actually just released quite a few preprints in the last couple months on various approaches that we have used to create these dropicles. I won't go into all of these in detail. I just wanted to kind of share that there's multiple ways to create these lab on a particle systems. Today, I'll be focusing mostly on this work that I spearheaded, which is using these particles for doing single cell secretion assays. So I kind of alluded to that we have these particles, we need them to be highly uniform, and we need them to have very distinct shapes. In this case, I argue we need to have some cavity to maintain a cell inside of. So how can we create these, and how can we create these uniformly and high throughput? So although I argue that we don't want to have to use special equipment or say microfluidics in the end assay, perhaps we can utilize it up front in order to fabricate materials that can then be distributed to other people. And so that's exactly what we did. So we used a microfluidic droplet generator. In this case, we're injecting a UV cross-linkable PEG polymer phase. With this, it's nice that it's very modifiable. You can add different reactive groups such as biotin, or say a peptide sequence such as RGD that allows for cell adhesion. In the other phase, we have a dextran polymer phase. So we mix these together, we generate droplets with these. And if you wait just a few seconds, they'll undergo phase separation. So these polymers are unique in that they're an aqueous two-phase system. And if you tune the polymer concentration just right, you can get this very distinct morphology where you have a outer PEG spherical drop and then a smaller spherical dextran component maintained within that larger droplet that's just barely touching the surface. Now, if you imagine you have these droplets with this morphology, if you excite them with UV light to cross-link the PEG phase, and then wash to remove the oil and transfer them back into water, you're left with these sort of bowl or kind of suspendable microparticle shaped or microwell shaped particles that have an exposed cavity. And the nice thing is you can create a lot of these particles in high throughput and then store them for later use. And the point I want to kind of emphasize here is that in terms of doing the assays that I will talk about further in this presentation, there will be no more microfluidics. So we are using the microfluidics up front in order to fabricate these smart biomaterials that we can then use as part of a assay that is compatible with just simple, common laboratory equipment. So great, how can we actually kind of meet the requirements that I set forth earlier? So the first thing is we need to be able to create uniform compartments. And the way we figured out to do this quite simply actually is if you take a suspension of these particles and you take some oil and surfactant, and if you just agitate it by pipetting, you can imagine that you will create smaller and smaller water and oil droplets until eventually you get to the size of the particles themselves. And what we have determined is that the droplets will actually stay at the size of the particles due to some energy minimization effects. And any excess fluids will be partitioned into these smaller satellite droplets. And so here's a bright field image kind of showing what this looks like experimentally. So you'll see these uniform, larger droplets that are formed with these particles. And you should see a sea of background satellite droplets. So this is really hard to see. So I'm actually going to go back to that original image that I showed. So here is a fluorescent image of the same thing. So here the particles are stained with a red dye, and there's a dye that is added into the water phase in order to help visualize exactly what's going on. So the first thing we see is that the droplets formed with these particles are highly uniform. We see that there remains an accessible cavity that holds a fluid volume that, again, is uniform. And although that we see some cases we get maybe doublets of these particles formed in a single droplet, when we look at the population as a whole, we see that these doublets are actually a quite small fraction of the population. And then if we look at the uniformity of the population, we see that all the droplets formed with these particles are very uniform. So CV diameter CVs of about 4%, which is comparable even with state-of-the-art droplet microfluidic systems. So great, we've met our first requirement. We can create uniform compartments, and we can do it with just a pipette with the simple common lab equipment. So how can we associate cells with these? So one thing we notice when working with these particles is that if you seed them into a well plate, you can kind of imagine that due to their morphology, they actually tend to settle with their cavities exposed upright. So exploiting this effect, what we found we could do is that you can seed the particles into just a normal well plate. Once they have settled and reached the bottom of the well plate, you can then seed cells in. In this case, we have particles that are modified with a integrin binding peptide sequence, RGD. We're using CHO cells as a kind of a test line. And if we seed the cells in and adjust the concentration, what we see is we can load cells into the particle cavities that they will attach. And if you tune the concentration of the cells, you can actually ensure that statistically you will have either one or fewer cells per particle. So in this case, we want to ensure that there's a single cell per particle. And when we looked at this over a range of concentrations, we see that this very closely follows Poisson loading statistics. Now, a keen observer will note that not all of the cells will end up in the particles, and that is an important point. We find that approximately 20% of the cells actually end up associating with the particles themselves. But actually, if we compare it to a lot of the state-of-the-art systems, such as droplet microfluidic systems that rely on using capture beads co-encapsulated with the droplets, that due to the double Poisson loading effects with these systems, they actually also have not perfect loading efficiencies. So this is actually somewhat comparable to the existing state-of-the-art systems and can definitely be improved on. So we can load our cells into these particles. They have these exposed cavities, which are great for holding them. What happens when we then try and emulsify them, so create dropicles with them? So what we see is that the cells actually stay well associated with the particles themselves. And what we also note is that over a significant duration of time, they actually stay fairly viable. In this case, the oils that we're using are oils and surfactants that have been well studied in the droplet microfluidic space and are well known to be biocompatible, have high oxygen transport. Another important thing to note too is that we rarely see cells that are attached to the outside of the particle, but we see cells that are very well maintained inside the particles. And our kind of ongoing theory is that the cavity itself acts as a shear shelter to protect the cells from any shear that's going on during the emulsification step, which could also provide benefits for improving the viability of the cells as well. So our cells are alive. So what happens if we want to recover them? Say we want to kind of isolate certain cells of interest. So what we can do actually is once we've emulsified them into oil, you can actually break the emulsion. So in this case, you use a mild biocompatible detergent to coalesce the drops and wash away the oil. And then when we recover the cells with the particles after this, what we see is that one, the cells still stay attached, and two, we can actually watch these cells grow over time. So in this case, we have a 24-hour, 48-hour, and 72-hour time point. You can see these like initial starting colonies are actually expanding over time. And at a certain point, they will actually overwhelm the particle and they will kind of engulf it and then spread onto the whatever surface that's below it. So in this case, they were seated in a well plate. And so they'll actually expand onto the well plate afterwards. And when we compare this with just seeding into a well plate as a control, we see that the relative growth of these cells is actually quite comparable. So great, we have a system that we can create compartments, we can keep our cells alive, we can recover them afterwards and expand them out. So how do we perform a functional secretion assay? So starting where we left off, we have our cells and our particles. And one unique thing about this system is that these particles aren't fixed. So you can actually resuspend them. So in this case, you can pipette them and recollect them into a conical tube. And you can imagine that while the cells are adhering to the particles, in this time, they might be secreting molecules, which will be creating a background signal. But uniquely with our system, we can actually just do simple centrifuging and washing steps to remove this background. We can then utilize some of the functionality of our particles. So in this case, we have biotin groups embedded into the particles themselves. We can add streptavidin, and then in this case, we're linking a biotinylated capture antibody that's specific to human IgG FC. Then once we do this, we can then immediately emulsify it into these dropicals. And so what this lets us do is it lets us compartmentalize the cell. Any secreted molecules in this case will then attach to the particle surface due to the capture antibodies. And then once we've incubated long enough to be able to get high enough signal, we can then transfer it back into water, as I discussed before. And then we can just label it with fluorescent secondary antibodies. And so what this kind of looks like experimentally, so here's a microscope image with a bright field image merged with some fluorescent images. So you'll see that with these are anti-IL-8 secreting CHO cell line. And what you'll see is that there are several particles that have single cells associated with them. And it's only these particles really that we see these fluorescent signal from the captured antibodies. And when we looked at the system, so kind of analyzing just with a microscope, some MATLAB code, that if we increase the incubation time, we see that the intensity of these fluorescent secondaries also increases with that time, indicating that one, we can actually generally quantify the amount of secreted molecules from these cells. But two, also it's very interesting in that we see that it's not a single peak. We see, it's not a very sharp peak. We see a very large distribution in the productivity of these cells, which is supposed to be a cell line, indicating that there is a lot of functional variability in these cells. So, kind of taking a step back, I think when we looked at the system, we really wanted to verify that this is working the way we think it we think it's working. And so, particularly, we were curious to see if we take the same system and put it into a bulk suspension. So in this case, we are not going to do any of the encapsulation steps and see what happens. And so when we perform the assay this way, what we see is that the particles with cells indeed have a fluorescent signal associated with them, which is expected. But kind of as expected as well, we see that a lot of particles without cells also have signal associated with them. And if you look at this over a population, what you'll see is that there is significant crosstalk. And there's an overlap in the signal from these populations, meaning that you're not able to really distinctly determine from a single cell resolution to quantify the amount of secretions being produced. So just doing this kind of same study again with the dropical system, we break, we wash, we label. We now see that only the particles with cells have signal. And then when we analyze this as a population as a whole, we see that there is, in this case, no crosstalk in the system. So great, we have a secretion assay. There is able to analyze single cells without crosstalk. And so the next part, as I discussed earlier on, is we want to be able to analyze as well as sort in high throughput with this system. So what we did to really kind of test this system even further is we took two different cell populations. So the antibody producing CHO cells that I showed before, as well as a cell line that does not produce any human antibodies. We labeled them with their own separate cell trackers so that we could kind of trace them through the experimental process. We then performed the assay as we had before. We did the labeling, and then we ran them through a commercial flow cytometer. And so what we saw when we did this is that, so this is an image of the pre-sorted sample. We see that the particles with the cells that should not be producing antibodies have, this is in this case blue, there is no signal associated with them. There's no green fluorescent signal from the antibodies. However, in this case, magenta colored stained cell, which is an antibody producing CHO cell, we do see a distinct signal associated with this. And when we when we ran this through a flow sorter and selected our population based off of the antibody fluorescence signal, we saw that we were actually able to isolate this subtype that we spiked in. And we looked at this over a range of different dilutions, so going from anywhere from 1 out of 5 cells containing the antibody producing CHO cells down to much more dilute cases of 1 to 1,000. And even in this much more dilute case, we still see that we're able to sort with a fairly high purity, above 80%, based off the secreted molecules. And I guess another important thing to note here too is for this last condition too, we had to screen over 100,000 cells within a single screen in a single day. So it is a very high throughput system. But this is really just kind of a control test to show that this assay and the sorting is behaving as we expect. We wanted to go a little bit further and see if we could do something maybe a little more interesting on a functional level. So what we did is we took this antibody producing CHO cell line, we performed the assay, and then we again ran it through a flow sorter, and we looked at the population of events with particles with cells and having positive antibody signal. And you'll notice if you kind of look at this top section of the graph that there's actually a sizable population of cells that are not producing antibody, so no longer secreting these molecules. And we noticed this earlier on too when we were doing microscopy analysis, and we did some further analysis to verify that this is not due to viability or things like that, but there's actually a subpopulation within our clonal cell line that is not effectively secreting the molecules of interest. So what we did is we selected 2 different populations, ones that are all producing, these are all producers. So these are the condition where we're just selecting out ones that are producing antibodies. And so we're trying to get rid of the population that's no longer producing. And we also took even a step further and selected out a higher tier of this subpopulation, so top 20% of producing cells. We then expanded these over a little over a week, and then we did a well-played ELISA assay to see what our production levels were. And so when we compare this with our pre-sort sample, we actually saw that in the case of the top 20% condition, we were able to achieve about a 50% increase in production. And this is with 4 biological samples replicated, so all performed in parallel. And we actually see for one of our subpopulations, we achieved about a 58% increase in the production levels. So it was great. So we were able to actually probe based off of a functional phenotype. So in this case, secreted molecules. We were able to sort based off that and actually show downstream that we're actually able to isolate a subpopulation of interest that is doing this. And this is all with this standard lab equipment. So I think a good point to bring up, I showed that slide earlier on with the various microfluidic systems. And my first main point was that, well, we want to create a system that is more accessible to researchers. So really emphasizing the fact that we want it to be compatible with any standard lab equipment. But how does the performance of our system actually stack up against these systems? Well, if we kind of consider these examples here, I'm just taking the amount of cells that you can screen per day as a metric. So for some of these cases, so microchambers and maybe optofluidic systems, the throughput is maybe a little bit less. There are other advantages to these systems, which I don't have time to go into detail about. Some of these systems too, the cell recovery, as I mentioned before, is quite difficult. So these microchambers and microwell arrays are not really well suited to sorting and high throughput. And another thing to maybe consider is the idea of cell compatibility. So a lot of these systems are theoretically compatible with suspension and adherent cells. With droplet microfluidic approaches, they are not very well suited for adherent cell types. As you can imagine, they have no surface to adhere to, and depending on how long you have to incubate the cells for, this is not an ideal situation. Now, if we compared it with our system, so our current system, we actually see that we can achieve throughputs that is comparable to the state-of-art microfluidic platforms. And part of the reason why that we can do this is due to the fact that we can analyze and sort using existing fax machines, flow cytometers. So these have been tuned over the last several decades to be very high throughput machines for analyzing micro objects or cells. The other thing too is that the way that we're forming these compartments is really massively done in parallel. It's not a serial approach. You're not manually pipetting single cells. You're not sequentially generating droplets in this case. So this allows us to parallelize a lot of the workflow. And if I kind of come back to this chart over to the right here with the cell compatibility, interestingly, our system currently is a little bit kind of opposite of these other droplet microfluidic platforms in that it's very well suited for adherent cell types. So currently we've been testing the system with cells that can adhere via RGD binding sites. But I think I really need to emphasize here that this is just really a first generation of the system. And some of our more recent work and what we kind of project in the near future is that if we address a couple of things, so making, say, particles smaller to be compatible with smaller nozzles on flow cytometers could increase the throughput of the analysis and sorting step. Improving the efficiency of loading could also significantly bump up our throughput capabilities for screening. And already we've been exploring other approaches to make this system more compatible with a larger variety of cell types. So whether it be B cells or T cells or other non-adherent cell types, and have had some limited, we've had some success so far, but I'm not going to go into too much detail. But we really see that this is a platform that can grow and can really even outperform all the existing state-of-the-art technologies. So just to kind of summarize what I discussed today. So we've created this platform that allows you to take this reagent, so a microparticle reagent that you can seed into a well plate and isolate single cells. You can form compartments with them by simple agitation steps, allow cells to secrete and capture these secretions onto the particles. You can then transfer these back into a water phase that is compatible with flow cytometers, label them with a fluorescent secondary antibody, and sort in high throughput. And really kind of just painting the vision for this platform and this technology, what we really see is we have a platform that allows us to take cells with interesting and functional phenotypes. We can use these microparticles. We can combine them with existing lab infrastructure and can use them to perform these very quantitative, high quality assays on single cells. And I think a really important thing to note too with this technology is that this is something that is very easy to distribute. So, I mean, already we're sending these particles to collaborators. It's very easy to ship. It's much different from having to create a altogether equipment or machine that you have to move and install into a new location. And the infrastructure we do use that's maybe more complex are already well established in existing labs. So testing out and adopting this technology, there really is a very low hurdle compared to other systems. I think this is actually really pertinent now where it's more difficult than ever to travel, where we're trying to limit interactions with others to kind of reduce the spread of COVID-19. And I think perhaps especially too with some of the applications of the system being particularly suited for potential therapies. So for example, for the production or isolation of cells producing neutralizing antibodies, this could really be a really powerful approach that many people could use. And kind of aside from these single cell secretion assays, we really don't see this as a kind of one-off tool. We really see this as just the beginning of a larger tool set that can be used to really probe the limits of biology. So looking at all single entities. So for example, looking at proteins at low concentrations or even single proteins using these microgel droplet ELISA assays, or perhaps looking at nucleic acid, so doing digital nucleic acid assays with these lab on a particle systems. And if I had to summarize this whole presentation in one slide, and maybe this is also particular for the lab automation folks, if you take your 1536 well plate, which handles fluid volume in the range of 1 to 10 microliters, if I could tell you that you could have a reagent system, a particle reagent system, that allows you to create compartments that are 1,000 times smaller, and that you can create more than 1,000 times more compartments, and you could sort using only existing lab infrastructure, what would you do with these systems? And on that note, I want to thank everyone who I worked with on this project, particularly for this work, Professor Dino DiCarlo, my lab mates, Robert DiMatteo, Mark Manzi, Professor Robert Namaso, one of our collaborators, as well as many others who have worked on other aspects of this project that I was not able to discuss in detail. But if you want to learn more about what we're working on, you can check out our website at biomicrofluidics.com. If you look on the bottom right, or if you want to reach out to me, you can always reach out to me on LinkedIn. And with that, I want to thank you all for your attention and all the best.